Many important discoveries in science happen by accident. Scientists may look for one thing, but they find something else entirely unexpected. This unexpected thing can be very useful. It might even change the world in big ways. This is what we call serendipity. It means a happy accident or a fortunate discovery. These moments show that science is not always a straight path. Sometimes the biggest breakthroughs come from surprising turns. Alexander Fleming's story is a perfect example of this kind of luck. These accidental findings are not just pure luck. The scientist must be ready to see them. They need a curious mind. They must notice when something is unusual. They must ask questions about why it happened. So while the event is an accident, recognizing its value is not. It takes a prepared person to understand what they have found. Many everyday things we use were found this way. From sticky notes to microwave ovens, accidents have played a part in their creation. This makes science exciting. The history of medicine is also filled with such stories. New drugs and treatments have often appeared by chance. Someone might be testing a chemical for one illness. They might find it works for a different illness instead. Or a side effect of a drug might turn out to be a helpful treatment. These events show that we must always be observant. We must be open to new ideas. Even a mistake can lead to something great if we pay close attention to the details. Alexander Fleming was a scientist working in London. He was studying bacteria. Bacteria are tiny living things that can cause illness. Fleming was known to be a bit untidy in his laboratory. This untidiness, however, would lead to one of the most important medical discoveries ever made. His story teaches us that sometimes, a little bit of mess can lead to a major breakthrough. It highlights how unexpected events can shape the future of medicine and save many lives. In the summer of 1928, Alexander Fleming went on vacation. He was a bacteriologist at St. Mary's Hospital in London. Before he left, he had many dishes in his lab. These dishes, called petri dishes, were used to grow bacteria. He was studying a type of bacteria called Staphylococci. These bacteria can cause infections like boils and sore throats. Fleming was known for not always cleaning up his workspace perfectly before leaving for holidays. This time, his habit would have amazing results. When Fleming returned to his lab in September, he found a pile of old petri dishes. He had left them by an open window. Some of them were contaminated. This means other things, like mold, had started to grow on them. This was not unusual in labs at that time. Many scientists would have just thrown these spoiled dishes away. They would have considered them failed experiments. But Fleming was a careful observer. He decided to look at them before discarding them all. As he sorted through the stack of dishes, one caught his eye. This particular dish had mold growing on it. The mold was a bluish-green color. But what was truly interesting was the area around the mold. The Staphylococci bacteria, which should have covered the whole dish, were not growing near the mold. There was a clear ring around the mold. It was as if something was stopping the bacteria from getting too close to this specific type of fungus. Fleming was puzzled and intrigued by this observation. He realized that this mold was not just a simple contamination. It seemed to be doing something very special. It was producing a substance that killed the bacteria. He thought this was very important. Most people would have seen only a ruined experiment. Fleming, however, saw something with great potential. He knew this was more than just a messy dish. It was the start of something big. Alexander Fleming took a closer look at the strange mold. He identified it as a rare type of penicillium. Specifically, it was Penicillium notatum. This mold had likely floated into his lab through the open window. It landed on the petri dish and started to grow. Fleming carefully took a sample of this mold. He wanted to study its effects more. He grew more of the mold in a liquid. He found that this liquid could kill many different types of harmful bacteria. He called the substance produced by the mold juice at first. Later, he named it penicillin. This was because it came from the Penicillium mold. Fleming did experiments to learn more about it. He found that penicillin was very powerful against bacteria. Even when it was diluted many times, it could still stop bacteria from growing. More importantly, it did not seem to harm human cells. This was a very exciting discovery. It meant penicillin could potentially be used to treat infections in people. Fleming published his findings in a scientific paper in 1929. He wrote about how penicillin could kill bacteria. He suggested it could be used as an antiseptic. An antiseptic is something that stops germs from growing. He also noted that it was difficult to produce large amounts of penicillin. It was also hard to keep it stable so it could be used as a medicine. For these reasons, the medical world did not immediately see its full importance. His discovery was interesting, but not yet a practical treatment. For many years, penicillin remained mostly a laboratory curiosity. Fleming continued his research, but he was not a chemist. He struggled to purify the penicillin. Purifying means separating the active substance from other things. Without a pure and stable form, it was hard to test it properly in humans. It would take more than a decade, and the work of other scientists, to turn Fleming's accidental observation into a life-saving drug. But the initial spark came from that one contaminated dish. The true power of penicillin was realized later, during World War II. Two other scientists, Howard Florey and Ernst Chain, took up Fleming's research. Along with their team at Oxford University, they found a way to purify penicillin. They also developed methods to produce it in larger quantities. This was a huge step forward. They were able to show that penicillin could cure serious bacterial infections in humans. This was a turning point for medicine and for the world. The impact of penicillin was enormous. Before penicillin, even a small scratch could lead to a deadly infection. Diseases like pneumonia, scarlet fever, and syphilis were often fatal. Doctors had very few effective treatments for bacterial illnesses. Penicillin changed all of that. It became the first true antibiotic. Antibiotics are medicines that fight bacterial infections. Suddenly, doctors had a powerful weapon. Many soldiers during the war were saved from infected wounds because of penicillin. It was called a miracle drug. After the war, penicillin became widely available to the public. It led to a revolution in how doctors treated diseases. Many illnesses that were once death sentences became treatable. Surgeries also became much safer. Doctors could use penicillin to prevent infections after operations. The discovery of penicillin opened the door for finding other antibiotics too. Scientists began searching for other molds and microbes that could produce similar bacteria-killing substances. This led to the development of many more life-saving drugs. Alexander Fleming's accidental discovery shows the importance of serendipity in science. His messy lab and his observant eye led to one of the greatest medical breakthroughs of the 20th century. While the mold grew by chance, Fleming's prepared mind recognized its significance. Penicillin has saved countless millions of lives since its discovery. It reminds us that sometimes, the most unexpected events can lead to the most wonderful outcomes. Science often advances through a combination of hard work, careful observation, and a little bit of good fortune.